Beginning, there is a muscle made up of fascicles, which are made up of bundles of myofibrils, which are made up of myofibers, or cells. Each myofibril has a nerve attached to it that sends its signals to contract. The removal of the signal allows the muscle to relax again. Contraction of a muscle starts with an action potential which travels down the axon of the neuron to the synaptic knob. Along the way, the AP opens calcium channels and calcium flows into the nerve. The calcium causes vesicles filled with the neurotransmitter acetylcholine to migrate to the end of the axon and release acetylcholine into the synaptic cleft. The acetylcholine flows across the synaptic cleft and binds with acetylcholine receptors on the motor end plate of the muscle. After this action is completed, acetylcholinesterase comes and removes the remaining acetylcholine. When the acetylcholine binds with its receptors, sodium is released into the muscle fibers, and it has effect depolarizing the membrane and continuing the AP along the sarcolemma. The sarcolemma has tubules that travel perpendicular to the sarcolemma, and the AP travels down these tubules and causes calcium to be released from the terminal cisternae on the either side of the sarcoplasmic reticulum.
Your hand.
The calcium binds with the troponin site on the actin filament. This binding process causes the tropomyosin to shift off the actin heads, which in relaxation are covered. As soon as the binding sites on the actin heads are uncovered, the energized heads of myosin, the thick filament, leaps to bind with the actin, forming a cross bridge. The myosin head pivots to the low energy state, releasing ADP and inorganic phosphate, creating a power stroke. That causes the thick and thin filaments of fibers to come together and contract the muscle. ATP from the mitochondrion then binds to the myosin head, causing the head to break free from the active site on the actin, and it becomes energized again as ATP is converted to ADP. The actual contracting unit is called a sarcomere. This unit consists of a Z-disc, I-band, A-band, H-band, and M-line. The sarcomere is defined by the area between two Z-discs. The I-band has only thin actin filaments. The A-band has both thin and thick filaments, and the H-band has only thick filaments. The M-line is the middle of the sarcomere. When the actin and myosin are bound together, the sarcomere is shortened as the muscle contracts and the filaments slide over each other. Meanwhile, since the action potential is no longer available to open calcium channels, calcium is pumped back into the terminal cisternae at the end of the sarcoplasmic reticulum and held there by calsequestrin. The calcium is no longer bound to the troponin, so the tropomyosin moves to cover the actin binding sites. The ATP pumps calcium back into the sarcoplasmic reticulum, and the muscle relaxes again.
Thank you, action potential.